Hello everyone, my name is Avantika and I come to you from Dr. Sarah Lutz's lab at University of Illinois, Chicago. And today I'll be talking to you about the blood-brain barrier and what happens to it during SARS-CoV-2 infection and long COVID, specifically how a protein called Caveolin-1 drives blood-brain barrier breakdown, neuroinflammation, and cognitive impairment in our mouse models. So the blood-brain barrier is the brain's gatekeeper. This is illustrated by Evan's blue staining here. In here, the Evan's blue dye was systemically injected, and as you can see from the images, that it floods the kidney and the liver, but it fails to permeate the brain. The blood-brain barrier normally excludes dye molecules, leukocytes, fibrinogen, complement factors, cytokines, pathogens, and most drugs. However, when it breaks down, the consequences are severe. So what makes the blood-brain barrier impermeable? And to understand this, we have to look at the structure of the barrier. It is made up of brain endothelial cells surrounded by pericytes and astrocytic end feet. And at the interface of different endothelial cells, there are structures known as tight junctions made up of claudin-5, ZO1 occluded that block the paracellular passage between the cells, and the endothelial cell cytoplasm itself maintains low transcytosis, limiting transcellular passage. Both of these routes become relevant during neuroinflammation. Here is the electron micrograph of a healthy cerebrovascular blood-brain barrier. And I hope that you can appreciate that the tight junctions are intact and there are low vesicles in the brain endothelial cells. When this vesicle-mediated transcytosis increases or the tight junctions open, there is barrier leakage. There are two routes by which molecules and cells breach the BBB. One is the paracellular gaps, that is through the endothelial cells when the tight junctions break. And the second one is the transcellular route directly through the endothelial cell body via vesicular transport. And I mentioned earlier that both of these are important during neuroinflammation, and a protein called Caveolin-1 is central to this transcellular route. There's a clear evidence of the blood-brain barrier breakdown in COVID-19. Gadolinium-enhanced MRI from COVID-19 patients shows areas of contrast enhancement, which is a radiological sign of the blood-brain barrier leakage. And post-mortem pathology reveals presence of fibrinogen, which is a blood protein that is not normally present in the brain, leaking into the olfactory bulb and the brainstem, co-localizing with vascular collagen. This indicates that the barrier is physically compromised. Accompanying this is the vascular damage in the CNS immune infiltration. There are regions of accumulation of CD8 T cells in the brain, along with activated microglia indicated by the IVO1 staining in the red. CNS T cell infiltration at this scale is not normal, and it reflects a significant blood-brain barrier failure and immune-mediated component of COVID-19 neuropathology. The overarching hypothesis in the lab is that cerebrovascular inflammation contributes to neurological effects of SARS-CoV-2 infection. And the timeline is very important here, right? The viral replication peaks and wanes. However, the host inflammatory responses can persist and amplify that in turn cause blood-brain barrier leakage, neuroinflammation, and functional cognitive impairment that outlast active infection, which we think is the underlying reason of long COVID. And long COVID is not a minor problem. It has been reported that approximately 2 to 11% of the US population reports long COVID symptoms, along with specifically the neurological symptoms that last up to 8 months, and that these patients never neurologically fully recover, or the recovery is incomplete or unpredictable. This is why it becomes important to understand the mechanism that drives this. And to understand this, we use a mouse-adapted SARS-CoV-2 in which there is a single nucleotide polymorphism in the receptor binding domain of the spike protein. This mutated virus binds to the mouse ACE2 receptor in the lungs, and the animals infected with this virus show a reduction in the body weight, indicating disease, and the survival rate in this mouse model is approximately 70%. And when we work with the SARS-CoV-2 virus, we work in a BSL-3 environment. And here is the team. Here is myself, Ali, Karisha, Remy, and I would so it's just all of us doing the virus work in the lab. We have two experimental models. One is the acute phase of the disease, and the second model is the long COVID. In here, we intranasally inoculate the mice with SARS-CoV-2 virus, and after performing behavioral assays, we perform euthanasia either at day 5 for the acute model and at day 30 or day 60 for the long COVID mice model. In the long COVID model, there is an initial reduction in the body weight in the acute phase of the disease, and the mice recover at the 30-day time point, and the survival rate is again approximately 70%. So the hypothesis for this particular project is that Caveolin-1 contributes to blood-brain barrier permeability and pathology in COVID-19 or long COVID, and that mice deficient in Caveolin-1 will be protected from acute and chronic signs of COVID-19, in part due to the protection of the blood-brain barrier. We then wanted to check if this blood-brain barrier permeability contributes to CNS T-cell infiltration, and we co-stained for CD3 and GLUT1 that marks the vasculature. In the wild-type animals that were infected with SARS-CoV-2, there was a significant increase in the amount of CD3 cells. However, in the Cav1 knockout animals, the number of T cells were comparable to the wild-type animals. We then wanted to see if this blood-brain barrier impairment and T cell infiltration correlates with learning and memory deficits. And we performed novel object recognition assays that has been quantified by discrimination index by discrimination index in the graph here. Discrimination index is the measure of the animals to discriminate between a novel object compared to the familiar one, while the wild-type animals showed a significant decrease in the discrimination index, indicating learning and memory impairment. The Cav1 knockout infected animals did not differ very significantly from their mock counterparts, indicating that Cav1 contributes to neurobehavioral deficits in association with blood-brain barrier permeability. Oh, and we also correlated this discrimination index with the amount of CD3 T cells, and there is a direct negative correlation between the CD3 cells and the discrimination index across individual animals that indicates that if there is more T cells in the brain, there is learning and memory deficits. We then moved on to the long COVID mouse model and wanted to see if wanted to look at the Caveolin-1 expression in the brain at 30 days post-infection. And for this, we performed flow cytometry and isolated brain endothelial cells gated on the CD31 positive cells that mask the brain endothelial cells and looked at the expression of Caveolin-1. And quantified in the graph here, the results show that there is a significant increase in the levels of Caveolin-1 at the 30-day time point. There is also a strong negative correlation between brain endothelial cells Cav1 and behavioral performance. Spanning mock and infected animals, that is high endothelial cell Cav1 predicts poor memory, even 1 month after the infection. And here is the behavioral assay readout, where we performed open field habituation and novel object recognition. At 30-day time point, the wild-type mice show significantly impaired NOR performance compared to the mock animals, and the Cav1 knockout infected mice maintain memory above chance and are not significantly different from the mock animals. Thus, Cav1 knockout protects from long COVID associated cognitive. We then wanted to look at the levels of T cells in the long COVID mouse model, and we performed flow cytometry to quantify the amount of CD3 and CD4 positive cells in the brains. Wild-type mice infected with the virus have significantly elevated CD3 and CD4 positive T cells in the brain, and the Cav1 knockout infected mice do not show significant elevation over mock in either population and are significantly lower than the wild-type infected animals. The protection from cognitive impairment tracks with the reduced lymphocytic infiltration. And at the structural level, we imaged for Claudin-5 at 30 days post-infection to quantify protrusions and tight junction gaps. And as you can see here from the quantification, that there is a significant increase in the protrusions and the tight junction gaps in the wild-type infected animals. However, in the Cav1 knockout animals, it's significantly lower. Thus, it indicates that Cav1 drives persistent structural blood-brain barrier damage at the long COVID time point. To summarize, SARS-CoV-2 infection drives Caveolin-1 upregulation in the brain endothelial cells. This increases transcytosis, breaks tight junctions, and facilitates T-cell infiltration into the CNS via both transcellular and paracellular routes. This results in neuroinflammation and cognitive impairment, both in the acute disease and persisting into the long COVID time frame. And removing Caveolin-1 blunts all of these responses, indicating less leakage, fewer infiltrating T cells, and preserved memory. And with this, I would like to thank and acknowledge members of the LUTS Lab, specifically Ali and Remy, that have moved this project forward. I would like to also thank the funding sources and our collaborators at UIC. Thank you. And with that, I can take any questions.